All right, so I'll be discussing the events at the neuromuscular junction. Here we have a motor neuron, and a muscle fiber. It intervenes. An action potential travels down the spinal cord to trigger the contraction of a skeletal muscle fiber. The action potential propagates down the motor neuron, and this signal travels down to the axon terminal of the nerve, and this entry of calcium causes the calcium channels to open, letting calcium ions into the axon terminal, as you can see here. This acetylcholine comes in contact with the synaptic vesicle, where it triggers the release of acetylcholine from the axon terminal into the synaptic cleft. The acetylcholine diffuses across the synaptic cleft and attaches to the acetylcholine receptors on the receiving postsynaptic muscle fiber. The acetylcholine binding opens ligand-gated cation channels here in green, letting sodium ions to come out the sodium ions to rush in, while the potassium rushes out of the muscle fiber, as shown here. So potassium here is the blue, it is coming out, and potassium and then sodium is going in flux inside the muscle. So as sodium rushes in and potassium moves out, it changes the electrical charge of the muscle fiber. This membrane potential, the change in the membrane potential becoming less negative due to the sodium influx and potassium outflux, creates an action potential down the sarcolemma. And when the charge reaches a certain level called the threshold, another electrical signal, also known as action potential, moves along the muscle's surface. At the end of this event, you see that the acetylcholine receptors are moving away from the synaptic cleft, and we have acetylcholinesterase enzyme breaking down the acetylcholine into acetic acid and choline. Choline is eventually recycled and it is recycled back into the nerve ending, where it is used to make more acetylcholine for future signals. The event that converts an action potential to a contraction is known as the excitation contraction coupling. In a single muscle fiber, an action potential propagates along the entire sarcolemma. Swiftly conducting into the interior of the muscle fiber, aided by the glowing transfer tubules, also known as T-tubules, which extend throughout the muscle fiber, as seen here. At various points where the T-tubules intersect with the sarcoplasmic reticulum, calcium, shown here in blue, is stored within the membrane. When the T-tubules comes in contact with the sarcoplasmic reticulum, it creates bulges resembling sacs known as terminal cisternae. The junction formed by the tubules encircled by the swollen sarcoplasmic reticulum is termed a triad. Proteins, shown here in pink, at the junction between the T-tubules and the terminal cisternae facilitate the connection of the two membranes, regulating the release of calcium from the sarcoplasmic reticulum. This protein changes the shape as an action potential travels down the T-tubules. This protein is voltage sensitive, which causes the protein to change shape. This change in shape triggers the release of calcium ions into the sarcoplasm. The swift increase in calcium levels initiates the contraction of the skeletal muscle fiber. Calcium ions play a crucial role in linking excitation to the contraction of skeletal muscle fibers.